Okay, I'm going to be going over the first worksheet that we will be using that's part of the GEP. I explained the GEP and some of the background in some other videos, along with some molecular biology, some simple things that probably the first week that we begin bioinformatics they need to be going through. The first thing that we'll be doing is we'll be using BLAST for our first assignment and in this worksheet. And I'm not going to go through the exact worksheet with you. In this particular worksheet, there's no answers for them to turn in. Let me phrase it that way. They're simply to work through it, and by working through it, they will gain the skills needed to do the next worksheet, in which there will be questions that they will need to ask and turn in. So the first week, we have a couple of worksheets. Some of them, they work through these kind of worksheets where you see it tells you where to go and you kind of click here, do this. It's very straightforward, but by doing this particular worksheet they will gain skills necessary to do the next worksheet, in which there will be actual answers. So I'm going to kind of walk you through BLAST, a very simple one. I think most of you have probably used BLAST. There might be some features that you're not aware of, so I will just get you started on some basic BLAST. I would suggest that you work through this worksheet yourself, but if you think you understand BLAST pretty well and you're short on time, maybe not, but the students will be working through this one. So, of course, if you're not familiar with BLAST, you can find it on Google. Through all of this. Alright, you can find it by just typing NCBI and BLAST. Now in the first worksheet they'll be using BLAST-N and TBLAST-X. If you're not familiar with those, I'll probably do a worksheet or a video or something where I go through and mention, kind of discuss the different types of blasts and when you would use one type and when you wouldn't use the others. But we're going to do BLAST-N, which stands for nucleotide, and I'll just go to the nucleotide BLAST and wait here. So this would be your typical BLAST page. Now notice up here at the top you can choose the different types of BLAST. BLAST-N, BLAST-P for protein, nucleotide, and it'll take some more explanation to do X. It involves translating back and forth and searching different databases. But for now, you can think of BLAST, if you're not familiar with it, as a search engine for sequences, either for nucleic acid or amino acid sequences. And depending on how you want your results to be searched and which databases you want to use, you alter the parameters within BLAST. So I'm going to go find a sequence here, and I'm going to put it in something I know something about. Now there is one that they'll be using in the GEP worksheet that is called the unknown FNA. FNA stands for it's just FASTA file, but it's an unknown sequence. It's large, and it will take them several minutes, maybe 20 minutes to search and get a result. I'm going to do a shorter sequence here, something that hopefully won't take nearly as long for us to search. And normally your search sequences are in the form of FASTA files, FNA or FA or FASTA. Those are all FASTA files, and inside those, of course, you can have either nucleic acid or amino acid sequence. Let's see here. So what I've done is this is the Homo sapien 7FL RNA. This is an RNA that is thought to be the precursor for the ALU element, the transposable element. It was mutated and learned to insert itself and reinsert itself in our genome. And this I'm just going to use this one as an example. It's not too big. Hopefully it will be found fairly quickly, and I won't be sitting here for 20 minutes waiting for it to give us a result. I am going to put a just test sequence A. That's good. You don't have to. Now, if you're comparing it to another sequence and you have another sequence directly that you want to compare it, you can click that, and what you'll get another place to put another sequence, and that can be very useful. So you might show the students that if you have two sequences you want to directly compare, you can click on that. I'm not going to do that. Of course, when you do that, you lose your the file you uploaded. So let's do this again. Let's find the... Alright, and we'll again call it test 1A. I won't click on that this time. I'll show you that a different time. Now, you want the nucleotide collection, perhaps, if you're searching all nucleotides. I don't want to search all nucleotides. In this particular case, I want to search Refseqs. Now, Refseqs, or reference sequences, are sequences that are used as references. They're kind of like wild type sequences. You might think of them that way. But they're the ones that, they're typically the first sequence for a gene that was actually sequenced. It may actually have no relation to the concept of wild type or not, but it's kind of thought of that way. But the first sequences that were sequenced ended up being the ones that they reference to compared to everything else. And it's probably the wrong way to think of it as a wild type, but it is considered in these databases the sequence in which other things are compared. So I'm going to choose the Refseqs, all right, and we can also choose down here an organism, and let's see, we're doing Drosophila. There we go. And so I can choose just Drosophila, so I can find something that's similar to this in Drosophila. Maybe they have something similar to our RNA. You might come back with their equivalent RNA to this. That's probably a very conserved sequence. What do we want to do here? Well, you can leave this blank usually, but if you do highly similar, it might take a while. You might get 0, as in you get no hits. Typically the one that works is somewhat similar, blast in, but you can tell the students to choose among these three and look to see which one looks like it's giving the more, shall we say, interesting results. It's like choosing a different algorithm in the browser to search. So you can have highly similar sequences, highly dissimilar, somewhat similar. In this particular case, I'm going to do somewhat similar. And I'm going to hit blast, and basically it's like a Google search engine. And we're going to wait here for a while, hopefully not 20 minutes, hopefully a very short period of time. And as you can see, there we go. Pretty quick. Well, let's see how it did. We scroll down and it tells us that in Drosophila, yes, in fact we did. We found the equivalent sequence in Drosophila. We found 2 hits. Now, how do we interpret these results? These are kind of like search engine results. Well, we interpret them by 2 things. Coverage, or percent identity, either or both of those, and the e-value. Now, the e-value with nucleotides is 1 × 10⁻⁶ or smaller. So this is -11. So yes, this is smaller than a 6 here. So 5th, sometimes some people choose to the -5th, others choose -6th. I usually go with the 6th, but it's up to you. Now, it's going to be much smaller number, much larger actually, negative. It's going to be to the -2nd or 3rd maybe when you do amino acids, but in nucleotides it's e to the -6th as a cutoff here for good quality alignments, and I'll show you what I mean by that in a second. Percentage identity and cover quality. Cover or query cover means how much of it did it actually match up. So these are certain size, and like what percentage actually kind of mapped. And then when you look at it, there is a percent identity. It's kind of like the percent similarity. So You usually go by the e-value. The e-value is kind of like the primary one, but they do give you a score to kind of mix these things and give you an overall result. Generally, I just look at the e-value. So let's look at what this looks like when you click on it. All right, and actually all it did was scroll down. But here is our alignment for that piece, and there are several pieces that are going to align. As you can see, we have a piece here, and you can see there are gaps. There are places it does not align, and there are pieces. So really, we only have two small pieces that align well, and it's enough. It's enough for the. This is non-random, or the e-value would be much larger, and so we would consider, okay, these probably are related in function and probably evolutionarily related as well, and you can begin to continue your analysis at this point. Now this is a graphic summary. Now this sometimes can be hard to explain to the students, but basically this is the region that we're looking at within our sequence that were similar. So they were similar in this latter region of the sequence. So if the sequence was roughly 300 here, it was between a little after 100 and not quite to 250 that they were similar. And the percentage or alignment score is 50 to 80. That means as a range, generally as you move along the sequence, about 50 to 80% of the nucleotides were the same, in the same order as well. So order is important too. There are, of course, gaps and deletions and insertions along the way, but as a general rule, this lined up, and we would consider this an above average alignment because of that e to the minus 11. That is a smaller number than e to the minus 6. So we would say that this is a good alignment. All right, now taxonomy, if this is interesting, it probably isn't interesting to us at all, but could be in other fields. And then, of course, we go back to the description. If you need to see about this gene, the one from Drosophila, we searched with a human RNA. Let's go find this. This will take us to... Hold on, it's loading. And it should give us the GenBank file here. Yes, now this is called a GenBank file. This is very useful to the students because this one may not be so much, but it often, yeah, here it is. It will give the sequence, but it will also give the start and stop places in a FASTA or in the gene where that this can be downloaded. In other words, the FASTA, if you don't know what a FASTA is, you can come up here. All right, you can hit FASTA here, or if you want to maybe download it, you can go File. And this was the GenBank, it's called the GenBank file. You can go FASTA, and you can create the file. All right, I'm going to take a look at it here. Now you do need to open FASTAs with a text editor. If you open it with something else, it can be altered by the like Word or whatever. So try to open them with Notepad in Windows. But this is a typical FASTA file, and this is the Drosophila signal recognition particle 7SL RNA. So this is an RNA. It's telling us it's an RNA. Now you'll notice in here that there are T's. That's because all FASTA files use T's. Even if you're talking RNA, it's going to put a T in here wherever the uracil was supposed to be. So just be aware of that. All FASTA files begin, all right, with a greater than sign and then a line where you can do a description, and then the sequence will begin here. And this, of course, is a nucleotide RNA FASTA, but you can also have amino acid RNAs. I mean, nucleic acid FASTAs. So there would be 20 letters. One letter that's often common in these, and not all programs can read, are N's. N's are unknowns. Just keep that in mind. If you see something like that in somebody's sequence and they don't know what to do, it's an N, and you can often take it out if the program you're using or whatever is not recognizing N's. All right, so this is a FASTA. This is what matched with, and I am going to copy this, and we're going to go backwards, and I'm going to show you how to do a different type of analysis. We'll go back to our original, and we're going to do the align 2. All right, now I'm going to paste the one I just copied in here. So I've pasted, cut and pasted, but this one I'm going to choose the same sequence that we used before, the Homo sapiens 7SL. Now the one we have in here, this is the human, and this is the fly that we just found. So here we're comparing the two, and I'm going to tell it to do highly similar. I did somewhat similar before, and we're going to just see what we get here. Now notice when we're doing two sequences, it doesn't ask us what species to search in or anything like that, because we're just searching between these two. We're analyzing, and it's an analysis between the two. Here I chose the file, here I cut and pasted the file, but we could have chosen or cut and pasted either of them. You can also take the subrange, like if I know that that region we were just talking about was like from 100 to 250, I could put in 100 and 250 here, and it would just compare on that range. So that's another trick you might show the students. So I'm going to leave it here, and we're going to search on this one. This should be relatively quick. All right, and oh, it didn't find any similarities. Okay, let's go back. It should have. I was expecting it to. Oh, here we go. Because we just did this type of analysis, but of course what did I do? I chose the top one, thinking that, oh, -11 should be more than enough to give us this, so I'm going to have to go back and choose the same one. That's really funny. -11 I thought would have been more than enough to give us an answer. Okay. We have it, and we now have compared those two, so we can get a graphic summary. Now this is showing you again what we already saw. It shows you the places between the two where they're similar, and we already knew that. We can also see an alignment. Again, that's the same as before. And we get something new and something very useful, which you do need to show them, which is a dot plot. And I will do a whole lecture on dot plots. But dot plots essentially tell you where the sequence is similar, and of course you have to set parameters, but of course it wasn't identical, but we see a solid straight line, and so the solid straight line is between 100 and 245. This zoomed in to the part that was similar, but there might be several dashed lines in here or some other lines which might indicate repeats and things of interest to us. But dot plots are extremely useful, especially when visualizing and trying to figure out how similar your sequence is, because sometimes it's difficult to look at something like this and interpret. Is that really that similar? You know, you get a number here. Look, here's a very low E value for just this piece. So it's telling us to just this piece. If we cut out all the rest of the sequence and just look at this small piece, this is an extraordinarily low number. That means it's extraordinarily similar. So this is considered very similar. All right, 0 would mean identical, by the way. All right, I thought I would show you just an introduction to BLAST for those of you who haven't seen BLAST. It also kind of introduces you to a GenBank file and to FASTAs. I have other videos if you have questions, or if the students do, on FASTAs and dot plots and GenBank files. So this will be our first assignment. Our first assignment here, we'll ask them to go through and do BLAST. They will use a couple of different types of BLAST. They'll use tBLASTx, and they will use BLASTn like we did. Here we go. We see in their worksheet something very similar to what we did. and they will be detecting homology. It walks them through exactly, and this is how most of these worksheets work. They walk them through everything, tell them exactly where to click, and they will get answers. They'll get numbers, and they'll ask them, well, what did you find for this? And so by working through these worksheets and answering them, they're going to be developing skills, and those skills then they can go back and use to annotate their own sequences, where they will be comparing their allele to, say, the wild type. That's their unknown versus the wild type, and finding out exactly the places that have mutated, and then commenting on, oh, we had a mutation here, and it was in the splicing site for this exon, for, say, the second exon and the first intron, say. And so therefore, by disrupting that, it changed the protein a great deal, and they can talk about that in their paper if that happened to be what happened. It might be just a plain simple single nucleotide polymorphism. If it is, they'll have to find it, and they'll use the skills that they gain here to find it and analyze the nature of it. So all of these things are here. Like I said, we learned about GenBank and FastAs. This little graphic here, this little worksheet, gives them a great deal. Now here you found more than just two. It found several. We were just comparing two things in ours. This is when you have many things, and this of course tells you that, oh look, this is conserved across many sequences, many species, for instance. This is probably an important part. These are the important parts. Maybe this part isn't that important. And that also is something they can comment on perhaps in WA5. So this is actually using Drosophila here. The example they use is for the mutation legless, and they will also be using some more other graphical tools here. This is part of the NCBI, and we did not look at that, and it's not necessary yet for us to worry about it. But I thought I would give you a quick run through of their first little assignment, which is to work through this sheet, getting ready for one where there will actually be questions associated with it. This is the taxonomy. We probably won't be looking at that at all, but they will learn how to do it in this worksheet. All right. This talks here about the fact that this was an mRNA, and for legless, and of course, what's their e-value? E-value of 0 means it's identical, so all of these are essentially the same sequence, and they're all in the database. Notice some of these are predicted instead of known. That means they were found in somebody sequenced a fly. It's a Drosophila. It's a homolog. So the sequence has been duplicated within the organism. Let's see, coding regions. Now they'll use BlastX, looking for more coding regions. This will be very similar to what we just did, except for that you won't be comparing it to another sequence. You will be in the Refseq. If I haven't told you about Refseq, remember Refseqs are the ones that are referenced in the as the sequence that all others are compared to. That is kind of important for them to know. All right, more alignments. Here, e-value of 0, important to note, that means identical. And here, with BlastX, you get results in amino acids. So here's amino acid sequence, and you can notice there are a few places where they're not the same. All right, this would be the kind of thing that they were looking for that would alter their phenotype. In other words, the things here, and if they can find places of importance that are altered, they can make a judgment call as to, oh, this must be what altered the phenotype. Actually, they can find that in papers if they look closely, and actually all of that information is in Flybase if they look hard enough. Again, a whole piece missing. And that should do it. Eventually we'll be doing intron-exon boundaries. They'll be introduced to this here. This is a slightly different tool. Actually works very well. and they will be just introduced to it. It gives this genome browser view. We will talk about that in a later video. And back to BLAST and more amino acids, and this will tell them, well, where does the alignment begin? And this is one of the things that they need to learn to do, is to interpret this and say, okay, here's the piece where the two sequences are lined up. In this sequence, it's from 1 to 60, and in this sequence, it's from 9,344 to 9,168. Oh, they can view their sequence here. And of course, this worksheet is designed so that they can do the next one, in which there are questions that they will need to ask.